Okay, friends, just to continue again, I know it's a hectic session from my side continuously, but I'll try to, detox you in between with a lot of breaks and polls and make it on the lighter side with a lot of interactive learnings for all of us across. So the next session from my side is going to be focusing on the essential, ensuring compliance with essential documentation practices for analytical R&D, analytical research and development, and the quality control. Now, the focus area here would be the numerous cases on the 483s, the drug recalls, the warning letters highlighted in the India pharmaceutical system, and the crucial to examine and improvement can be made across with the best practices. Let me put my pointer probably with the best practices for the documentation. Now, while the quality document at every instance is very vital, we all know that, but R&D has to work very, very closely with the quality team, whether it's QC or QA. So this session will help you enlighten how the best practices for maintaining the accurate and comprehensive documentation is very, very critical for, essential documentation practices for both analytical R&D and the QC. And I will take you through the knowledge transfer model, which I specially devised for this presentation for all of you to learn across there. So I'll take you through this session. Okay, so 483s. I know Form 483s is a known fact for all of you. When the US FDA comes for an audit, they issue a Form 483 based on observations, which are critical, major, minors. So I'll not go through the definitions, straight to the database. So year 2024, which is immediately the last year, how many 483 observations were issued from the FDA in the system? So if you look at there are several categories, biologics, devices, drugs, food, human tissues, radiological, veterinary. I'm just focusing on the drugs part here. So around 561 483s were issued only for drugs in one single year, which is 2024 by US FDA. So this shows the grave numbers which have been issued only to the drugs, which are the pharmaceutical drugs, the ANDA filings or the ANDA commercialized products. So this table does not represent the complete 483s, but they were evaluated based on the manual database from the US FDA website, and they have a complete spreadsheet also available. If somebody is interested, they can go to this FDA database site, which the reference has been given. So they cite across all these observations in a very comprehensive manner. Total 483s were 4,056, out of which 561 were relevant to the drug products only. Now, what are the different types of drug, 483s which were issued in 2024? So I've just highlighted few in the red ones. Procedures were not written or fully followed. Very, very critical. What does it mean? As per the FDA database, the responsibility and procedures applicable to the quality control unit, so G, again, QC is more critical here, are not writing fully, and there is a detailed description to it. And there are 201 observations out of this which are directly related to the procedures not being written fully. So how critical is good documentation practices, not only in QC? This is showing across the 483s issued in 2024 by the FDA. Investigation and discrepancy failures. So there's a failure to thoroughly review the discrepancy, meeting the specification for the drug product, which has already been distributed in the market. So the deviation or the OS or the OT was not adequately justified in the root cause analysis, FIMs, FMEAs, and agency was not satisfied with the way the investigation was completed without finding a root cause. This contributed around 134 483s. Absence of written procedures, which I was just talking a few minutes back. The firm failed to establish written procedures for production, whether they're SOPs, the MBRs, manufacturing batch records, packaging batch records, process controls, to assure that the drug product have identity, strength, purity, and quality that they're purported or represented to possess. So this possibly is also contributing as a major contributing factor for 483s. You see the numbers is 126 in the 1 year itself. Then another major cause which was coming in the top 4 was scientifically sound laboratory controls. Laboratory controls not only include scientifically sound justification, specification, sampling plans, or test procedures, and also contains the logs for the drug product containers, labeling, conforming to the appropriate standard of, again, identity, strength, and quality. So what is the meaning that we are getting? The summary we are getting across here is somewhere, yes, manufacturing procedures, good documentation practices, OS, OT investigations, the knowledge transfer happening from analytical R&D to quality control unit, or to quality assurance units from R&D is very, very critical and leading to many 483 observations, which we need to look into, review, evaluate, and minimize going forward. Going next in the top, 5 categories, equipment design, cleaning, sanitizing, maintenance is one of them. Procedures for sterile drug products, yes, that's also contributing. Written procedures not established or followed. Somebody, Shikha, asked the question previously. So written procedures are not established for cleaning or maintenance of the equipments, whether they are manufacturing equipments, whether they are lab equipments, utensils for manufacturing, processing, packaging, holding of drug products, even sometimes the glassware, contamination happening across in the QC lab or leading to a lot of OS results, whether in impurity profile or other investigations, leading to around 60 numbers of 483s. Computer control of the master formula records, this is another contributing factors. So if you look at the trend data, I just try to categorize them across, in the trend data. I've highlighted few of them in the blue ones. I'll focus on them. Obviously, you can go through the remaining ones also. Investigation or discrepancies or failures. The failure of any batch or the component which was already distributed was not adequately justified, was not meeting the specification limits, like it was a OS or a OT or a deviation, which was the highest contributing factor. Absence of written procedures, whether it's in the QC lab or in the manufacturing area, leading to not having the confidence in the identity, strength, purity, and quality of the product. Scientifically sound lab quality control procedures. Again, lack of the written stability programs. Then going down, following the documenting or laboratory controls. Test methods, doing the accuracy, sensitivity, specificity, reproducibility of the test methods as per the ICH. So method validation was not available for the QC before they release the finished product into the commercial market. Also led to 483 observations. Complete test data was not included in the records, So the complete data, like the tests, the examinations, the essay necessary to have the compliance for the established specifications was not available, either for the standards or for the samples. So this was also contributing. Approving or rejecting the components or the products which were, not meeting the specifications, but they were still approved. No written record of investigation. Somewhere the investigation was not even documented. They just did a repeat testing and released the product into the market, which is not acceptable to US FDA. It's a serious concern with respect to, 483 observations nowadays. It's taken very seriously. SOP is not followed or documented. They don't have a deviation management SOP or a written procedure or SOP for good documentation practices, how to do an investigation, lab investigation report, OS, OTs. So those procedures or SOPs were not available. Complete test data like laboratory records were deficient, was not available during the testing part. They were missing data records in the lab from either QC. And procedure deviations, recording and justifications was not properly justified either in the production area or in the quality control unit. Deviations were not properly justified without a proper root cause, and they were completed and closed without adequate justification and releasing the product in the markets. So these are some of the major ones, short description and long description, all available in the US FDA database. I try to give you a flavor of why these 483s are getting, coming across, where they're coming, and how we can strengthen our analytical R&D to quality control, knowledge transfer model to minimize the number of 483s and such kind of observations. So again, these are again the top, 10 483 observations. I will not go through them again, probably. But again, scientifically sound laboratory controls, as for the subpart I, what is subpart I? Subpart I is laboratory controls. So if you see that appropriate specifications, standard procedures needed an improvement. So this had around 84 observations out of that. So all these are given across, or the subcategories are given across for your reference purpose. Again, nature click. I go into the forest areas, tracking, mountains, and I found beautiful flowers. You see with the dew droplets, it's a bulb-shaped flower, it's a kind of a lamp, but it's a unique flower I found in Taiwan during my tracking inside the mountains. Just to share with you all, again, a digital detox for all of you. And yeah. Getting more of the deep dive into the inadequate procedures part, because we found that is critical, attribute for the 483 observations and for the corrective actions to be taken. Inadequate procedure refers to procedure which are not adequate or appropriate to ensure the compliance for the FDA regulations. For example, failure to establish and implement written procedures for quality control, testing procedures for the finished product, lacking the procedures for control and handling of the raw materials. So even the API, the excipient release, the packaging material release, they also lack procedures and effective controls, not only just the finished product. Inadequate procedures for maintaining and calibration of the equipment used in the manufacturing process, in the laboratory units. So probably those records of the equipments, whether used for manufacturing and for the release of the finished products, are also proved to be inadequate. So all these account for inadequate procedures as per the 483 observations. I just give you a very example, unique example. If you go to the USP monograph of Montelukast oral granules, this is how the sample solution preparation is documented. And I would like to thank Shailesh for this, case study he shared across with me. I put across in this presentation. So it says normally 0.24 milligram per ml of Montelukast is prepared. Transfer 60 milligram of Montelukast from the packets into 500 volumetric flask. Add 250 ml diluent. Shake well. Sonicate 30 minutes. Occasional shaking. Filter through 0.45 micron filter. Centrifuge. Clear solution. Finish. What is missing? Now, if I go into the deep dive of this part, a lot of things are not documented across, which have to be mentioned in the quality control or from the analytical R&D when they do the method development. They have to ensure the same procedure. If I put across, what are the missing elements? The detailed sample transfer process from the sachet to the volumetric flask with pictures has to be identified and documented in the monograph, which has to be transferred to the quality control unit so that they understand how the sachet has to be cut open, how the content have to be filled into the or transferred into the volumetric flask with appropriate pictures. Detailed procedure for shake well, because here it says shake well. But what do you mean by shake well? A strong man or a weak man, what do you understand by it? It's a subjective term. So we have to define the intermittent shaking. I would say every 5 minutes. You have to do intermittent shaking 10 times, so that the finished product and the diluent are adequately mixed and we get adequate accuracy or recovery in the finished product when we are doing the assay or other testing. It says sonicate for 30 minutes. But the critical attributes which are missing out here is, what is the sonication bath temperature? 25 degree, 30 degree, 40 degree. It can lead to thermal degradations. So we have to define that very clearly in our finished product monographs. Make up the volume and mix well. How to do that? Keep up for X time. After making up the volume, you have to allow some time to allow the sediments to come to the bottom of the volumetric flasks. So what is the time has to be defined. So all these critical attributes, the analytical critical attributes which have been defined, discussed in the AQBD part in the morning sessions also have to be documented in the monograph, very clearly defined for the QC to follow. Include the procedure for the discard volume. It says just discard. How much? We have to do a filter discard study. 1 ml, 2 ml, 3 ml, 5 ml, and basis that the optimal discard volume has to be documented in the finished product monograph or the API monograph, to how much of the optimal volume of discard has to be there from the filter before it is filtered into the HPLC vial. Specify the type of filter. It says just 0.45 micron, but you have to do a filter study whether PVDF filter, nylon filter, or any other alternate filter can be used for filtering the sample and the standard solutions. It says centrifuge. But what is the tube size? What is the rotation per minute? What is the temperature? 3000 RPM at 25 degrees Celsius, tube size of 10 ml. All these critical attributes have to be defined in the procedure monographs, which are currently missing across there in the USP procedure. Last but not the least, maybe the last two, include all the sample preparation related precautions in detail. If any, photolytic degradation is there, so use amber color volumetric flask. Temperature controls, environmental controls, all they have to be documented in the finished product test procedure monograph. Sample storage temperature, whether it has to be kept below 25 degrees Celsius. What is the stability in analytical solutions? It is stable for 1 day, 5 hours, 10 hours at 5 degrees Celsius or at the room temperature. So all those critical attributes have to be defined across there. So this case study I typically takes across to make you understand that if I need to make a monograph for Montelukast sodium oral granules finished product, I need to take into account a lot. Apart from the USP procedure, I need to take into account all these critical attributes also. Good documentation practices. Another reason for, I saw in the 483s and, in the previous slides. So poor record keeping. The examples can be Like FDA found that agency or the organization failed to maintain accurate, complete, up-to-date records, whether for manufacturing, for testing, and distribution activities, which includes maintaining records for the equipment maintenance and calibration. So qualification records have to be ensured with the quality team across, whether it's DQA or the quality team unit function, quality assurance. Incomplete or inaccurate record of manufacturing and testing activities, lab notebooks, logbooks, MBRs, PBRs. Failure to retain records for the updated required time period. Data retention and retrieval policy is very important as per FDA requirements. All these are a part of good documentation practices to ensure that record keeping is up to the expectation mark. Effective trainings. We saw that effective training is must have for a successful knowledge transfer and for a effective batch release. So lack of employee training was one of the root cause which we saw in the previous example, which was leading to non-compliance with respect to regulations. Example of lack of employee training can lead to failure to provide the trainings on handling and storage of whether raw materials, finished product, how to do the sampling, at what temperature, what RH controls are required, any nitrogen gas purging is required or not. So all those critical attributes have to be provided in the training program for API, for finished product, or for the packaging material. Lack of training on the quality control procedures, Alcoa plus, I don't need to go through that part maybe. So all those training protocols have to be followed for the QC control procedures defined in the testing methods. And insufficient training on the safety procedures or the use of equipments related to EHS and qualification have also to be ensured when you are doing effective training programs for your GMP facilities or for manufacturing and for the quality control units also. Another critical attribute or reason behind the recalls and also 483 observations was contamination and cross-contamination. So we saw that, inadequate cleaning procedures, lack of hygiene, improper handling of raw material finished product led to contamination and cross-contamination. So this also included presence of bacteria or microorganism in the finished product, leading into failing in the sterility or, the PET test, contamination of product with the cleaning agents or other chemicals, cross-contamination of product leading to improper handling due to the improper handling of the raw materials. So all these contribute or they have to be taken into consideration to avoid any kind of contamination or cross-contamination when you're doing a batch testing and a batch release across for the GMP batches. Next critical attribute was failure to follow the written procedures. So following the written procedure, if you don't follow them across, this leads to non-compliance and the production of the products that do not meet the FDA standards. Obviously, they have to be recalled and that leads to 483 observations. So failure to follow the written procedure for cleaning, sanitization of the equipment, the cleaning validation is a must-have, following the cleaning method validation, the cleaning procedure, the cleaning sampling and release for the release of the equipment. Failure to follow the written procedure for quality control of the finished product. The monograph has to be well defined. Example of Montello cast granules, how the monograph has to be written with all the precautions and AQBD attributes. And failure to follow the written procedure for handling and storage of the raw material, the SOP part, the trainings. What are the consequences of Form 483 and warnings? It can lead to delay in the approval of the new products. FDA may uphold the approval for a medical device or a drug product if the observations are there. And this can lead to loss in revenue for the organization and market share. It can lead to suspension or revocation of licenses also as a serious consequences. When 483 or warning letters are delivered, then it can suspend, the FDA can suspend and revoke the company's license to manufacture or distribute the products in the future. Legal actions can be taken by FDA if the company has violated the FDA regulations, leading to fines, penalties, and legal consequences, and it leads to the damage of the company reputations, negative publicity leading to violations, and this can erode the consumer confidence in the company's products and decline in the sales. So the 5 best practices for responding to the 483 observation: understand the observations first, very critically. Investigate the findings using the adequate tools, FMEA, 5 Whys, Fishbone. Review the past QC, GMP, and R&D trend data, very important. There are a lot of things which come from the research and development database, that what was the historical data which could have led to this OS or OT specification, out of specification results. The document coming from the API vendor DMF to correlate with the QC data with it's related to the API, and develop a corrective and preventive action plan. CAPA plan is a must-have, and responding to FDA with a follow-up and preparing for the future inspections. So defining the root cause, implementing the solutions, verifying the solution works, and all coming from the research or R&D. So this is all integrated across their, in continuous model. Fishbone diagram, again, root cause analysis, machine, method, measurement, material, manpower, mother nature, all can contribute into failure, OS, 483 observation, drug recalls, and investigations in the GMP lab or the quality control units and the manufacturing units. So we can always use this tool, Ishikawa diagram, to find out the root cause. Cross-contamination, I think this we have already covered in the previous presentation. So this is again an example how the contaminated products are getting released, finding out the real most probable root cause, fixing up the root cause, having a CAPA in place, implementing the CAPA and making that CAPA effective. Check has to be there across so that CAPA effectiveness check is provided before we close the CAPA procedure. CAPA is very important as per ICH Q10. A structured approach is provided in ICH Q10 to the investigation process used with objectives to determining the root cause, and the level of effort, formality, documentation, investigation should consummate with the level of risk in line with ICH Q9. So you see, CAPA takes into consideration R&D findings, adverse events, MDRs, complaint, process failures, a lot of deviation management. So implementing a successful CAPA includes standardized procedures, resource allocation, trainings, communications, tracking and trending. What are the benefits coming from CAPA? It leads to improved product quality, enhanced product efficiency, reduce risk, continuous improvement, customer satisfaction. Nature Click, again, digital detox, beautiful ocean with the 7 arches, bridge is there. And then you can go to this island. You can do a mountain tracking across on this island. So ocean, bridges, island tracking, all in one picture along with the sky and the landscape. This again island space in Taiwan, which I just captured for information and just to take a digital detox off. Okay, how to avoid receiving the Form 483s and warning letters. Now this is most important. Analytical R&D to quality control, the knowledge transfer model, which is a very important attribute. Detailed analytical test procedures, which we covered in Monte Lucas, how they have to be put across, documented, precautions documented, minimize. This will help minimize the QC related investigations, the number of investigations, the reoccurrence also. Also, we should conduct regular self-audit and assessment for the continuous improvement program, for the Kaizen approach. Potential improvements for the non-compliance with the FDA regulations. Companies should regularly review their procedures, their SOPs, their monograph, and their training programs all across. Employees should be trained in all relevant FDA regulations. Companies should have system in place so that records are accurate, complete, and up-to-date as per ALCOA Plus requirements. And audit trail obviously is uneditable in the real-time basis because a lot of observations are coming based on the data integrity issues recently based on the electronic audits happening across by US FDA. All QC investigations to have detailed investigations, having the defined root cause analysis, effective CAPA in place. So all these have to come across from analytical R&D function going into a quality control unit so that this leads to a complete knowledge transfer model. I'll go into more details of this model. So the knowledge transfer model will include addressing all the inadequate documentation practices, any common pitfalls which are there in the documentation, whether in the monograph, specifications, documents, training records or SOPs, and it will help implement strategies to maintain a comprehensive and a compliant record in the GMP lab. Also, this also will help us across in enhancing the deviation investigation process. If people understand the root cause analysis, the CAPA management process, this will help related to the inadequate deviation investigations and minimize the number of 483 observations which are linked to that. And this will improve the deviation investigation process happening across in the QC unit, all support coming from analytical R&D to find out the real root cause, whether it's coming from man, material, machine, measurement, or mother nature. There are several other things which I'm not touching like the change control systems, but I'm just looking into the major critical attributes like ensuring the data integrity so that the quality control unit understands the concern and observation related to data integrity for it, CFR Part 21, Part 11 compliance, implement best practices to ensure data integrity in the regulated environments in the GMP laboratory, having their own, password and access controls available. Proactive preventive strategies to be implemented across with proactive approach to ensure prevent common observations based on the database we have from the FDA. We can obviously have a learning from those database and prevent those FDA observations when an audit is triggered off from their side. and foster a culture of compliance within the organization with a continuous improvement and a Kaizen approach. This is just a snapshot of USP 1224, which is analytical method transfer workflow, starting from the transfer unit to the receiving unit, what packages are taken across, whether it's in a collaborative, direct method transfers, then execution of those, or a co-validation model. So all those workflows have been just put in a workflow so that we understand that effective knowledge transfer model includes an effective analytical method transfer happening from analytical R&D to a quality control unit or to a transfer unit, so that the knowledge and the procedures are effectively transferred from the R&D stage into the QC stage. So effective AMT procedure has to be followed with a documented signed off protocol and report, and all the precautions being listed across in the analytical method transfer certificate. Knowledge transfer model. Now, this is something which I've already implemented in our setup or regulatory compliance environments. Whenever an R&D transfers the knowledge into a QC lab, they have to do a micro level knowledge transfer model has to be followed. So which includes all the parameters, just taking the HPLC method. What is the pharmacopoeial status? USP, EP, BP, JP, others related to the assay, degradation product, dissolution, brain informity, other techniques. Similarly, what are the precautions which are related to the mobile phase preparation? What is the mobile phase stability? How stable is your mobile phase? Is there any precipitation observed after a few days? What is the sample form and sensitivity to light, moisture, thermal degradations? What is the diluent selection studies we have followed across? How the optimum diluent was selected? So that QC understand all these critical attributes for the procedures or the test monograph, and they effectively follow that in day-to-day batch testing and release. Sample transfer process from the sachet into the volumetric flask. Same example, Montelukast. Method precautions, take into consideration the analytical quality by designs. Sonication time, temperature, filter study, SIS, all taken into consideration. Speed, time, temperature. Sample extraction study, so that we have adequate recovery and accuracy coming across with mixing well, dilution, sedimentation rate, light precautions, all taken into considerations. Sonication time, intermittent shaking, solution stability both for sample and the standard, and filter compatibility and the discard volume. So all these knowledge attributes have to be transferred from analytical R&D for each of the test method like assay, RS, dissolution, brain uniformity, and other tests like residue solvents, so that QC understand all the precautions, critical attributes, and they implement that not only in the procedure monograph but in the training sessions also. Continuing further, the chromatographic parameters transfers is also important across there. The instrument model to be used, the column type to be used, the column temperature, why it is critical, flow rate. Obviously, the robustness study is done, but all these transfers are also done. What is the kind of needle wash to be used? System suitability parameters, RF, validation data, including specificity, accuracy, linearity, precision, LOD, LOQ, obviously. So all those have to be transferred to the QC lab. Obviously, there will be a complete validation being done in a GMP lab environment, but a partial validation data from analytical R&D is a must-have. So the critical attributes and the database is also coming from the analytical R&D to QC in the knowledge transfer model. Use of validated Excel sheets, multimedia dissolution data, representative chromatograms as an example for assay, impurity, dissolution, so that QC understands how is the peak pattern, peak elution, RRTs, and peak separation. Stability data trending from R&D stability data. All those knowledge transfer has to be done to the QC so that they understand what is the impurity profile increase or trending across in assay or the dissolution profile. Is there any cross linkage observed? So all those attributes have to be coming across from the analytical R&D to the quality control unit during the knowledge transfer model. And these are all based on the practical life experiences which I'm sharing across with you. So the practical tips for this analytical R&D to QC is proper training. Obviously, you have to ensure that all the people are trained on the methodology part, procedures, validation reports, and the personals who are doing the analytical testing in the GMP lab environment. All the methods which analytical R&D is making or developing are simple and reproducible methods. They understand the objective, the scope. This provides a complete clarity in the transfer process from all the transfer unit to the receiving unit. All the methods should be QC-centric approach should be followed. Why I'm saying so? Because if you have too much of complex methods, for example, derivatization, sometimes they would be too tough to handle at the QC lab sometimes. So if you can avoid such complex methodologies or sample preparation techniques, that will be easy to absorb in the QC, and less of investigations or failures will be observed in the QC or the GMP lab environment. Effective communications. So the sending and transferring units have to put everything in a documented communications, training programs, in the methodologies, procedures. And also we have to follow a proactive risk management approach to ensure that there are no future failures or OS or OTs or deviation triggered off in the receiving laboratory. The last slide before I conclude the session and open for the Q&A. So this is how, the method procedure, analytical development is a starting point. You develop an analytical procedure, method procedure, validate, improvement in the methods, and finally achieving the ATPs, and then, giving the feedback. The whole feedback cycle is a continuous one. So minimizing the kind of investigations, failures, OS, OTs, and deviations across there. So the methods should be user-friendly analytical methods. They should use a simplified approaches, reduce the testing time, whether sample preparation or the impurity profile testing methods should not be too long. Then method should be cost efficient with risk mitigation strategy in place, should ensure that regulatory compliance is ensured. They should be reproducible, robust with reliability in the test methods. They should be scalable and adaptable for the analytical methods in different labs, whether happening at a CMO site or a quality control unit at a GMP lab. So just to conclude, the process of developing the QC-friendly analytical methods, you have to understand the product from the R&D perspective, selection of the right analytical method by the analytical R&D, identifying the CQAs, validating the analytical methods, continuous monitoring and improvement, and analytical method transfer, leading to a successful knowledge transfer model across to QC, leading to batch release and minimizing the risk of any kind of observations or 483s or OT and OS and deviations across there. So with that, I come to an end of my presentation. Thanks for your patience. I open for the Q&A session. Thanks a lot.
Thanks a lot, Parminder ji, for that power-packed session, starting from analyzing past drug recalls and then moving forward to essential documentation practices for analytical R&D and QC. I mean, this was amazing, like back-to-back sessions with your inputs, and you're going back to detox and then coming back to the session. That was pretty amazing. So I'm sure there are a lot of questions. Now, one question we have from the previous session. Okay, so we'll go to that first and then we'll move ahead. So the question is, what would you do if a compendial method fails verification in your lab?
Very good question, Dharminder. If a compendial method, for example, I take an example of finished product, fails or is not able to, implement that method, a pharmacopoeial method as per USP, because the formulation design or the formulation excipient types might be slightly different for the test batch, which may not be actually possible. It may fail in specificity, accuracy. So it is possible that you can always tweak around the pharmacopoeial or the compendial method with a due justification, rational, why it is applicable to the finished product design, why maybe it's not a Q1, Q2 formulation, and why that modified methodology, which is non-compendial now, has to be subsequently validated. And with a due justification, a validation report has to be submitted to the regulatory agency of why we are not finding or why we're not implementing a compendial method. But yes, a non-compendial method can still be implemented and used across for testing and release of a finished product dosage form in case you find that your pharmacopoeial methods are not workable in that case. But has to be supported with a due justification and with a validation report across there in the regulatory filing why this method has to be implemented. Yeah, it's possible.
Okay, amazing. So we'll now move on to the next question. And this is on this session which you've taken on documentation. So the question is, training on documentation is given to quality but not R&D. Mostly we don't know what is expected. There is no cross collaboration encouraged. So how can the mindset be changed?
Good deep dive question, I would say in this case. See, documentation training has to be implemented across not only in the quality control, quality assurance, or manufacturing unit. It has to be done start from the basics where the method starts or the manufacturing design starts, the formulation design starts. So just in case a mindset change, what the question is talking about is, yes, Analytical quality by design is the first step where the analytical R&D understand what are the critical attributes for understanding the critical attributes for a finished product, dosage form. How do we implement a QC friendly methods? What are the critical attributes with respect to, robustness trials? And those thresholds are to be understood. And all those procedures are adequately implemented and documented there across in the test monograph, in the specifications, in your CMC documents. If you make a, dissolution development report, for example, I'm just taking a case study. So all those critical attributes are well documented across in the dissolution development report or your test method development reports, so that all that critical attributes are transferred from the analytical R&D into the QC lab in format of a adequately procedurized, defined test monograph and specifications, and all knowledge transfer is happening. So you're right, absolutely. If the mindset is not there, it has to start from the basic root cause. The management has to understand that training is must have, has to start from the basics, from the R&D. If they don't define a suitably defined procedure or documented procedure, it will lead to a lot of investigations, failures, recalls, 483 observations in the quality control of the GMP unit. So we have to start from the basics. That's why I started this knowledge transfer model. Has to start when the R&D understands the basic requirements of a thorough documentation, and they transfer this knowledge into the GMP labs.
All right, thank you so much for answering that question again. Now we'll move on to the next question. And it's, as per CEP 2.0, is it not required to perform method verification for EP pharmacopoeial methods? How companies are aligning with this requirement?
So Shikha, as per the EMA requirements, if you say across there, CEP, if there's a method validation available, report available, or it's a compendial method coming from European pharmacopoeia and you're going for a Europe market, they don't ask for a method verification by the ANDA filer. But on the contrary, if you go for US FDA, even if you have a compendial method, they ask you for a method validation for raw material, for excipients, and for finished product as per the recent FDA expectations. But you're right, the CEP, requirements, if the method is already validated by your DMF holder, by your API supplier, then in the European market or the EMA does not ask for a revalidation by the drug manufacturer. They only ask for a sometime, do kind of verification study, but they don't ask as a part of regulatory or a compliance requirement as a part of the filing package. So you can avoid that across, but I would still recommend as an internal confidence building exercise, we should still ensure that the compendial methods are workable for our, because sometimes the API sources are different, coming from different route of synthesis, different impurity, different residue solvents. So sometimes the compendial method or the CP versions are not effectively, practically workable for all kind of API sources across there. So it's still recommended that you still verify those procedures internally in-house to get that confidence. But for regulatory filing and compliance, they are not a must-have.
All right, once again, thank you for answering that question. We will now move on to the next question. And it mentions a lot of common mistakes in past and present. Using case studies from the past is good practice, but with frequently changing guidelines, same mistakes happen because of different reasons. And how to fix it?
Okay. The question is slightly very maybe broad, I would say, not clearly defining, but I'll still try to answer that to the best of the information provided. So it says there are some common mistakes in the past and present. Yes, there are 483 learnings and coming across from the the observations coming from deviation management, drug recalls, and the case studies. Yes, provides a good practice, but guidelines. See, the regulatory agencies always keep on improving. The ICH guidelines. US FDA requirements, the EMA requirements, like the nitrosamine requirements. So you have to always keep on updating yourself or enriching yourself with the recent guidelines and requirements so that you follow the most updated guideline, because agency always give a time period to implement that guideline. It doesn't say you follow tomorrow. Even if you are given some time period for review, you can always go back to agency. If you find some concerns, you can give that feedback mechanism. But I would recommend that we should keep ourselves updated with respect to all ICH, FDA, EMA guidelines and the global agencies, whether it's to Canada, JP, and other markets, and follow those guidelines so that we are not doing a non-compliance, or when the agency comes for a regulatory audit on site at the manufacturing site or the QC site, they don't find them as a 483 observations. So it's highly recommended. We always keep ourselves updated with respect to all guidelines and implement them in a time-bound manner for all our manufacturing process, QC related process, and GMP release also.
All right, perfect. So we'll now move on to the next question, and it mentions how can we be preparing for a filing like one planned for August 2025?
Okay, there's a complete CMC checklist available for the regulatory requirements, different modules available. So I'm not sure you are coming from a regulatory perspective or analytical perspective, but just to give a flavor to you, if you're filing for a August 2025 filing, if it's a US filing, you have to be available with a 6 month stability data, ACC CRT, and intermediate if required for US market. And then you have to have all the method validation package available. You have to have all the CMC package, multimedia dissolution profiles available from the analytical package I'm talking about. You have to have the drug dissolution development report available. You have to have the comparison between the R&D and the test batches available. So all that comes as a module, obviously along with the API validation, finished product validation package. So all these come as an analytical package, getting ready yourself along with the stability data for the August 2025 filing. But along with that, there are different modules with respect to the batch manufacturing records, packaging batch records, and all those PDRs, product development report. So it comes as a complete CMC module package from a regulatory perspective. And there's a complete guideline for that, which has to be ensured if there's a filing coming up in August 2025.
All right. Thanks to you once again, Parminder ji. That was an amazing session. And thank you for answering all our questions and taking it up. So we have actually come to the end of the session and with the conclusion of the Q&A. Would like to thank you once again, sir.
Thank you all. And I would thank the EBM team, Guneet, Dheeraj, Abhay, and Sally, and the entire team in the backend who had made this interactive session really wonderful. I really appreciate your team efforts, Guneet, and your team has done a wonderful job. Thanks for that.
Thank you. Thank you very much. So this is why I thought that I'll just come back on the stage and say that again. I really enjoyed your session. It was really beautiful. The way you have put a longer time in a use in a way that none of the people were actually getting dangled about the fact that it's a long conversation. It was a very important session, and a lot of people have obviously submitted some questions anonymously as well. As you see that we have shared a WhatsApp number as well with people, that if somebody wants to ask a question, and we have got a lot of questions anonymously as well. And once again, thank you very much, Parminder. Wonderful session indeed.
My pleasure. Thanks you all, and most welcome. You can have those questions shared with me across. I'll be glad to respond
100%.
Definitely, yeah.